Good afternoon, everyone. I'm Natalie Piperidis, and I'm delighted to extend a warm welcome to all of you joining us today for the last SRA webinar of 2025. First of all, I would like to begin by acknowledging the Aboriginal and Torres Strait Islander people of this nation. We acknowledge the traditional custodian of the lands on which SRA is located and where we conduct our business. We pay our respects to ancestors and elders, past and present. Today's presentation will run for about 40 minutes, followed by the Q&A session. And today we are in for a treat, and we have combined two presentations with two brilliant entomologists from SRA. So firstly, I would like to introduce Dr. Emtia Chandrima, who will give us a research update on Sugarcane Soldier Flies, a major pest of sugarcane in many growing districts. Dr. Emtia Chandrima has over 17 years of international experience in applied entomology across academia and industry. She specialized in sustainable and eco-friendly insect pest management. Her current work focuses on management of key pests such as cane grub, moth borer, soldier flies, major insect vectors in the sugarcane industry. I'm actually asked Emtia if she was not an entomologist, what she will be doing, and I think she will be a traveling YouTuber. So, and I said I'll come with her too. Our second presentation will be on future management of Australian sugarcane beetle and will be presented by Dr. Samuel Bawa. Dr. Samuel Bawa is an entomologist research scientist. He joined SRA Moringa in 2020, where he works on cane grubs and soldier flies. His research focuses on insect chemical ecology, applied entomology, and IPM. His passion lies in translating scientific discovery into real-world solution. I think I know entomology is a calling, and when you are an entomologist, it's a lifetime commitment. Actually, I personally know some entomologists which used to work at SRA and are still very deeply involved in entomology. So I also asked Samuel if he was not an entomologist, what he would be doing, and I think he will be a businessman. So now, after this brief introduction, I'm going to hand up the virtual floor to Emtia. Thank you, Emtia.
Good afternoon to everybody. My name is Emtia Chandrima. I'm working as an entomologist for Sugar Research Australia, and I'm going to talk on Sugarcane Soldier Flies research updates. Introduction. Soldier flies are an important economic pest that are found from far north Queensland to the New South Wales border. Now, the larvae of sugarcane soldier fly can feed on the cane roots, damage the roots, make the root system weakened, resulting in poor rattoning, and cause economic yield loss. This economic yield loss, not only from the yield loss, but also this economic loss is due to the plow out of premature blocks. Now, both these pictures show you that how poor rattoning can be occurred due to soldier fly infestation. As you can see here and here, there are no ratoons, and this is due to soldier fly infestation. And here in this image, you can see that due to poor rattoning, the space is occupied with weeds due to soldier fly infestation. Now, in the field, both the soldier fly and pupae are found, which are camouflaged well in the soil. So very, very hard to detect them in the field. So we actually wash them. And after washing, we can look at in the microscope like this. Now, let's look at the life stages of soldier fly. There are 4 life stages. Number 1 is the egg. Egg is hatched into larva, and the larva feeds on the roots. So larva is the destructive phase for sugarcane. So larva becomes pupa, and now the pupa is an inactive stage. It does not feed on any sugarcane roots. Pupa then emerge into adults. Now the adult flies, they only live for a very short time. But within this short time, they can lay up to 400 eggs. I'm talking about the female adult fly. So this is how the life cycle continues. So this is a general picture of how their life cycle proceeds. As you can see, the fly, they lay eggs, and these eggs are hatched, and the incubation takes place 3 weeks. And after that, the larvae are formed, and this larvae can go up to 9 months, all through here. And then the larvae become pupae for 3 to 4 weeks. As you can see that most of the life cycle, more than 90%, is actually the larvae. However, there is a twist, and that is soldier flies have complex life cycle of up to 2 years. So in case of 2 years, the larvae, instead of 9 months, can live up to 18 months. And in that case, we might have small larvae in winter and larger one in summer. So in addition to this complex life cycle, the other challenges for soldier fly management, number one, they are patchy in field distribution. Unlike cane grub, which requires a high pressure of cane grubs to cause significant damage, but in case of soldier flies, only low number of them can cause significant damage. They are unpredictable in terms of outbreak. So from one region to another region, their outbreak differs. And there is relatively uncharacterized varietal selection. That means if one variety is badly affected or affected in one region, that variety might not be affected at all by soldier flies in another region. In addition to this, there is species importance. Previously, 2 economically important species have been reported, Inopus flavus, Inopus rubriceps, but focus was mostly on the management of Inopus rubriceps, as you can see in this picture. However, the distribution and economic importance of these species are not fully described. In terms of management, many insecticide and variety field trials have been completed, but no recommendation. Only cultural control is recommended by SRA. But why no recommendation? It might be that there are more than 2 species that are functional across different regions. So based on these variability in terms of insecticide and variety field trials, lack of knowledge on distribution and economic importance, lack of knowledge on how many species actually are there functional, there was a need to do a research on soldier fly. And that's how we started a 2-year research project on soldier fly. And we followed these 3 major components. The number 1 was soldier fly distribution and damage. At first, phone survey was conducted to get information on damage and varieties, and followed by the ground survey. There were 70 growers across different regions with high response rate during the telephone survey. During the ground survey, we collected different stages, such as the larvae, pupae, and adult from different regions, and identified them through morphological or taxonomical way, and also through molecular. And finally, we also tried to develop the artificial dye system to rear soldier fly in the laboratory for a rapid screening of insecticides, biorationals, and gene products in the future. So let's look at each of the components' result. First is a telephone survey. This table is based on growers' perceptions during a phone survey. Varieties were reported as either badly affected or least affected on their farms. Here, badly affected is termed as highly susceptible, and least affected are termed as relatively tolerant. As you can see, the bold-fonted varieties appeared in both categories. For example, Q208 was found in FNQ in both the categories, badly affected, again, least affected. Again, some varieties, as you can see here, was often badly affected in one region, but least affected in another region. For example, Q228 was found badly affected in southern region, but not for FNQ. So these show that there is no specific varietal characterization. It might be because multiple soldier fly species are causing this kind of inconsistencies. Now, let's look at the next table that's on soldier fly distribution. Now, through the phone survey, we could found out that the smaller area of far north Queensland was damaged, like 20%. But when we did our ground survey, we found some blocks were severely affected. And again, in the central and southern regions, the effect was kind of relative. In some farms, it was like 25%, but for some farms, it was up to 100%. So you can see here we have reported far north Queensland, and this is the first report that is showing us that far north Queensland can also be affected, and we collected more than 1,000 samples from there. So from these whole broad range of varieties, we sampled very close to 3,000 soldier flies during our survey. Now, how did we collect or sample this soldier fly? This is how we did it. On the left-hand side are the larvae and pupae sampling in the field. And for the adults, we used YOLO pan trap to collect them. And these are the stages of larvae and pupae sampling across different regions. Now, let's look at the species identification. Now, there were two methods we followed for species identification. Number one was morphological identification of adults. We used the field collected adults, as well as we reared the pupae, and when the pupae emerged from the pupal case as adult, as you see in here, we then identified them with an expert taxonomist morphologically. Then to back it up, we also did molecular analysis on the larvae and adults. And for that, we did DNA sequence of the mitochondrial cytochrome oxidase I gene, amplified it by polymerase chain reaction, and sequenced them, followed by phylogenetic trees generated using multiple models. And larvae were selected for molecular analysis, which were on similar sizes. Now, first, with the morphological identification results, as you can see on the top half, showing already known two species, Inopus rubriceps, Inopus flavus, in Innispell, Mackay, Bundaberg, but for flavus, it was only in Mackay. However, there were two putative new species that were found. It's called Inopus rubriceps SPN4, 171 in number, from Innispell, Bundaberg, and Mackay. And for Inopus flavus SPN3, it's a new putative species was found from also in Mackay, along with Inopus flavus, the well-known species. But how did the taxonomists identify them morphologically? Let's look at that. So these were the identified species. On the left-hand side, you can see the well-known Inopus rubriceps. But if you look at this one, very similar to the Inopus rubriceps, but its veins have colored, like black color venation, with the anterior margin of the wings have smoky appearance, which differs from this one. And this is a putative new species. Again, for this one, Inopus flavus and Unipus flavus species N3, we can see the difference is only on the thorax, where there is no stripe on the thorax, but here the new putative species has 2 bands on the thorax. Now, as we're calling it putative new species, what is putative means here? Putative means they are suspected to be a new species, but they're not yet formally confirmed or described. So this is how the expert taxonomist morphologically identified 4 species. Now, for molecular analysis. Well, this is how our molecular analysis looks like. This is a preliminary molecular analysis, and more in-depth analysis is needed. Here, the phylogenetic tree is like a family tree showing several clusters. Each cluster is called a clade. Here, a cluster is called a clade. Like this, there are many clusters, but what is a clade? A clade is a group of individuals that are closely related and come from the same ancestor. In this tree, each clade over here represents a separate genetic group of soldier flies. So as you can see, in the left-hand side, there is one clade, and right-hand side, there are several clades, and each of the clade has individuals, and that individual's DNA was sequenced, and then this phylogenetic tree was generated. Now, as this left-hand side and right-hand side has many clusters and clades, so it's difficult to get very good understanding from this phylogenetic tree. So we will separate them in the next slides. But if you look at this tree, you can understand that there are several clades. That means there are several separate genetic groups of soldier flies across different regions. So let's break them region-wise. First is the far north region, where we can see that it had Atherton clades that is circled in brown and blue, followed by Innisfil clade circled in pink. Also, you can see in the tree. It is the first time that Innisfil clade has been reported. So after the far north clades, let's look at the Burdekin clade. We found there was one clade, that means one genetic group in Burdekin. Now, let's look at Central clade. Central clade is surrounding the Mackay region, and we can see there were 2 clades. One circled in orange and another one is in yellow color. And these orange colored clade is a major clade found in Mackay. So that means there are 2 genetic groups in Mackay. What about in Southern clade? Southern clade give us 3 in number that are circled in red, purple, and in light and green. So there are 3 genetic groups in Southern clade. What about New South Wales? Only from 8 samples of adults, we got a distinct clade in New South Wales. So in total, there were 9 clades, meaning there are 9 separate genetic population or genetic groups across different regions from 451 larvae and 30 adults. And as I mentioned earlier, this is a preliminary molecular analysis. So more in-depth analysis is needed to be done with more adult samples. So as we talked about the species, now let's look at our final component. And it was the artificial diets for bioassay development. We developed for the first time artificial diet that mimics sugarcane root chemistry, as sugarcane soldier fly feed on sugarcane roots. But why we need artificial diets? Artificial diets are needed because for stable and sanitized laboratory system for rearing soldier fly larvae. So with larvae available in controlled condition, we can rapidly test any insecticide, biorational, and future gene-related products. And this is far more efficient than any direct field trial, because direct field trials are slower, and then there are variations, such as variations for climate, or you can have unhealthy soldier fly population in the field, which can give you inaccurate results. And that's why artificial dyes very important for rearing the soldier fly in the laboratory. And we use two formulations of artificial diet. One was the solid diet, as you can see in this picture, followed by a liquid diet. A liquid diet, only a small drop you can see here, and both from the sugarcane root chemistry mimicked. And then we also used a control. That means it had no diet, no food. So we prepared our diets like this, and weekly we checked the survival of the larvae from each of the diet to evaluate the performance of these diets. Now let's look at the results for that. This is the results, the x-axis showing the time and the y-axis the larval survival in percentage. And you can see evidently both the solid and the liquid diets performing much better than that of the control or no diet. And there was a high survival rate until day 35 with the solid and liquid diets. However, in the control or no food diet, you can see that the population declined rapidly, and also, the survival declined rapidly, and there was 0% survival by day 21 with no food or diet. So this tells us that both solid and liquid artificial diets are very important for a healthy and stable laboratory colony of soldier flies. So in conclusion, what we can say, that we have found that all regions, starting from far north Queensland, central, southern, then also Burdekin, all these regions showed that they are severely affected with soldier flies, but not New South Wales. Why is that? It might be that we had limited surveys in New South Wales. And then if we have a further survey, we can conclude on New South Wales. Then morphologically, we identified that there were 4 species, including 2 putative new species. In terms of molecular analysis, we can say that multiple genetic groups are present across different regions. So soldier fly population are regionally structured. So that means there is no single or uniform population of soldier flies all over the regions. Both artificial diets, like solid and liquid, are critical in development of a laboratory rearing system. So if we continue on all these components and further research are done on this, then there should be an enhancement of the future control of soldier fly. For the future directions, even one species of soldier fly can cause great economic loss. So if we have more than one species in a region, that should cause more economic loss. So in future, we want to do further surveys in some regions, such as in Mackay, in Bundaberg, where there are complex and dynamic population of soldier flies present to complete the genetic diversity and to determine the key species in each region. Knowing the key species, we will use them for artificial diet bioassay to screen the potential control agents. Again, we want to use these key species to conduct species-specific varietal screening. And at the end, the extension of such research in future will ultimately lead to a sustainable management of soldier flies, which is profitable for the industry. I would like to thank SRA and Department of Primary Industries for funding this project, and also to Iris Laboratory, Brisbane, and Dr. Brian Lissard from Canberra, and also to all the growers who helped us enormously during the project. Thank you very much for listening.
Thank you, Emtia. That was a great presentation, very clear, very informative, and I'm sure there will be like a lot of questions, but we're going to proceed with the second presentation from Samuel, and then we will hold the Q&A session. So Shamso, hold your question for the end. Thank you. Thank you again, Emtia. And Samuel, the virtual floor is yours.
Okay, thank you so much, and thank you for coming. My name is Samuel Bawa. I'm going to talk on future management of Australian cane beetles. Cane grubs, which are the larval stage of cane beetles, are a major insect pest of sugarcane in Australia. There are 20 different species that attack sugarcane, 19 of which are endemic. Although there are 20 species, the most damaging and widely distributed species is greyback. This is the most damaging and widely distributed species, greyback. Well, because of the fact that life cycle plays a very important role in insect management, there's the need to understand the life cycle of greyback. The life cycle of greyback is made up of 4 life stages. That is the egg, the larva, the pupa, and the adult. What happens is that the adult, which feeds and breeds on various trees after breeding, will fly into the cane paddock, lay their eggs in the soil. The eggs incubate for a few days, then hatch into the larva. The larva goes through 3 developmental stages, i.e. first instar, second instar, and third instar. The third instar, therefore, develops into the pupa, and the pupa develops into adult, and the cycle continues. The larva stage is the destructive stage, and that is the stage that lives in the soil and feeds on the cane roots. By doing that, it deprives the plant of nutrients and water, therefore leading to yield losses. This is currently what we do. This is the stage that is targeted for control. That's the larva stage. And you can see from this slide, destruction begins from the late instar stage. These are some of the damage symptoms of greyback, and if not controlled, can cause about $50 million losses annually. How do we control cane grub? Over decades, control has mainly relied on the use of a single active chemical insecticide, which targets the larva stage. Although these actives have been very successful, but they come with environmental effects. These actives are currently under review by the APVMA and are likely to be withdrawn or have restricted use. It is therefore important to look for alternative management approaches targeting a different life stage like the adults. And one way to do this is by the use of volatile organic compounds. So therefore, the question is, what are volatile organic compounds? Volatile organic compounds are compounds released from different parts of plants. They can be released from the roots, the leaves, the fruits, and the flowers. They can either, when they are released, they can either attract or repel. So insects use volatile organic compounds to locate their host plants, even when they are covered with other plants. In the US, host plants, volatile organic compounds have been used to control or manage Japanese beetle, green June beetle. So because of this, we are hopeful and we think this could potentially be used for our Australian cane management. This is currently what we do and where we are. We want to do this or go here in the future using volatile organic compounds instead of using chemical insecticides. So our aim, although this project is a 1-year pilot project, which has 2 aims. 1, to develop methods that can accurately collect plant volatiles both in the field and in the lab. And 2, to identify volatile profiles of 3 host plants. This table shows the plants or the host plants that we used. We have strangle fig, sandpaper fig, and sugarcane. These plants play a very important role in the population dynamics of grayback because they serve as feeding and breeding trees. What happens is that when grayback emerge from the soil, they will first have to aggregate. They will find these host plants, aggregate on them, feed, breed, then fly back into the soil and lay their eggs. So, how did we collect our volatiles? We collected the volatile using a dynamic headspace volatile collection system, which combined SPME and sorbent. We collected the volatiles from intact branches with leaves that were enclosed in oven bags and securely sealed. We pushed in filtered air into the oven bags, and the outgoing air was pulled through a sorbent trap. So this trap will absorb whatever chemicals are in the headspace. At the same time, we push in or we insert a fiber, an SPME fiber, through the wall of the bag, and the sampling is done simultaneously with the adsorbent. This slide shows some of the collections that were done as a collection process. So what did we do? After collecting, our samples were analyzed using a gas chromatography mass spectrometry. Peaks were used to determine or to detect the compounds. So each peak here, each single peak here is a compound. And the compounds were identified using the NIST library. So what did we find? A total of 63 compounds belonging to 3 groups were identified. The groups are green leaf volatiles, aromatic compounds, and terpenoids. You can see from this table that the volatile profiles of the plants differ. Whilst strangle fig emitted a lot of sesquiterpenes, sandpaper emitted more monoterpenes. This group contains a lot of the long-range volatile organic compounds. So, what are the individual compounds that we found? Like I said earlier, although we collected 63 compounds, for the sake of this presentation, I could not display all the compounds. So what I actually did is to look at the compounds that were present in the most attractive plants and absent in the less attractive plants, since we want to first identify and see which ones can in the future be used to develop attractants. So this table shows two groups of compounds, and it can be seen here that both sandpaper and strangle fig did share two green leaf volatile organic compounds as well as one aromatic compound, however, in different amounts. As strangle fig emitted high amount of this compound, sandpaper emitted low amount of it. However, both species emitted this compound, which is 3-hexanone, in equal proportions. All these compounds that were present in these two species were absent in sugarcane. And the good thing is these compounds have been linked or have been associated to insect attraction and also signaling properties. On the group of terpenoids, which is monoterpenes and sesquiterpenes, it can also be seen that both fig species did share a lot of monoterpenes and a few sesquiterpenes, but these were absent in sugarcane. So what it means here is, or what we are anticipating, or what we feel, or what we think is that the presence of these compounds, which have been linked to be attracting insects and have signaling properties, may be the reason fig species are highly attractive to cane beetle, that is grayback, than sugarcane. So these are additional compounds of sesquiterpenes that were produced by strangle fig, but were absent in sandpaper fig. Of course, strangle fig is highly attractive and highly attractive to grayback compared to sugarcane. And the presence of all these compounds are linked to insect attraction and signaling ability. However, they were absent in sandpaper but present in sugarcane, present in strangle fig. And these compounds could be the reason, or maybe contributing, or could be contributing to the high attraction of grayback to strangle fig. So in conclusion, we have designed an apparatus that can accurately capture volatile organic compounds in both field and laboratory conditions. We have also identified the volatile profiles of 3 host plants of grayback. And we have shown that the volatiles of the two most attractive host plants, are different from the less attractive plant. We have also found out that the volatiles of the fig species, emitted a broader volatile profile than sugarcane. Although we have identified potential attractant compounds which may be used to develop attractants for cane beetles, there will still need to be further research to ascertain or to confirm that. So in the future, what we intend to do is to develop prototype layers which will incorporate our candidate compound and will test them against grayback. We'll also test them in the field depending on their performance in the lab. Based on the data that we'll get, we'll look for a commercial partner to develop for us a product. Then, once that is done, our future management option, we are going to target the adult beetle, and by so doing, we are going to reduce population pressure and also reduce insecticide usage. I would like to acknowledge SRA for funding this work, and also want to thank the growers who gave us their facilities or properties for the work. Actually, this work was done in Cairns and the Burdekin, but the data for this presentation is for only the Burdekin data. The Cairns data was excluded. Thank you. And if you want to see a full list of the various compounds that were collected, you could refer to this paper that contains a detailed and a full list of all the compounds that we collected. Thank you.
Thank you, Samuel, and thank you, Emtia. It was a very informative session today. I have to say that is like really fascinating work. So, but I think like for me, there's a bit like too much field work. So a lot of snaked. So it is now time for the questions.
In your presentation, you mentioned that there is a variation of infestation of the soldier fly within same variety, and it's actually happening in the same region and also in between different region. So my question is, and then your assumption was that it is due to the species differences. But why have you ever considered that it could be due to the environmental differences? Because you said the soil chemistry or maybe the seasonal variation may be involved.
Thank you very much for your question. It's an interesting one. Yes, of course, there are a lot of factors. One of the factors, as we have seen, the genetic differences across different regions. So that means the genetic difference, different genetic groups across different regions. It occurs due to difference, it could be due to adaptation to the climate, the soil, to the plant type, all these, and there might be this kind of genetic population that occurred. That can happen. So the species differences came from all these abiotic factors as well. But the species is one of the major factors, has to be. As you can see, we have got 9 clades. That means 9 different genetic population that are across different regions. And so when we look at the varieties, if in one region, one variety is bad for soldier flies infestation, but another region, it's not, because that region maybe doesn't have that species, and that species can maybe tolerate, that species is not, that plant is susceptible or not susceptible to that species. So there is a species-specific screening of varieties required to actually answer this question, and that's why I put that in my future plan. If you look at it at the end, that we wanted to do a species-specific screening of varieties, because as far as I know, so far, no varieties was shown to have any kind of tolerance against soldier flies. So we need to do that at the end of maybe with the further research. So yes, of course, the factors that you said, important, as well as the species difference that also arise from these factors. I hope that answered the question.
Thank you. Actually, I think you just answered my second question as well, because you already doing this. So I guess the reason actually we're saying that, because nematode may be slightly related to insect, but we found actually we have about 4 or 5 species of root lesion nematode. Only one nematode is actually causing the most damages, but the other, they are not that important. So that's the reason. Yeah. Okay. Thank you so much.
Thank you. Thank you.
So, but Emtia, like I see like Q208, for example, in like different region is like either like highly susceptible or tolerant. So I'm just not really understand
Yeah.
So there's no resistance like from Q208 like at all.
Actually, this table is based on the perception of the growers. While we were doing the telephone survey, we asked them that in your farm, do you have badly affected variety? And if they say yes, we count them as badly affected. But in the same region, the other grower might say, no, this variety is not badly affected in my farm. So based on that, we made this table. And as we said, at the end of this table, as I said, that there is no specific characterization to say, well, this is a badly affected variety, this is a tolerant or not that much badly affected by soldier fly infestation. So that's why we need the species-specific varietal screening once we know the key species across different regions. So our work was a baseline information or background information study. So now that we know there are multiple genetic groups, the next would be how we can identify the key species from these genetic groups and then go forward from there.
Yeah. So I just have a following question. So when you're going to do like some trial, would you actually add some like wild species like the spontaneum to see if they are more resistant or yeah, have more resistance to soldier fly than cultivars?
Oh, that's one of our plans there. But as you can see from my last slide on the future plans, first is we have to determine what are the key species, because now we have genetic clades, separate genetic groups. So to know if the genetic groups means different species, then we have to think from there that how we're going to plan our varietal screening. But first stop is our key species determination. From there, for species-specific screening, we might have to do as you said, but it's not has been finalized yet.
Right. Thank you. Okay, so I'm just staying with you, Emtia. There's a question from Gary.
Hi, Emtia, great work. So you did a morphological analysis that identified 4 different species, including the 2 new ones.
Yes.
Did you do the molecular analyses on those? And I'm just interested how those how the different species you identified worked into your phylogenetic analysis and in your geographical analysis.
Thank you very much for that question. It is interesting, actually. Yeah, well, of course, the morphological identified species were then the same, from the same region, the same species then went on for the molecular analysis. But the problem with the morphological and molecular is that morphologically only the adults can be identified. And we didn't have many adult samples, especially from the central and the southern regions. Adults were not so many. But in terms of molecular analysis, we could do both on the larvae and adults. Larvae were 451. So obviously we got more, I would say, wider breadth of results than that of morphological identification. In terms of geographical location, actually, we just wanted to just break down the clades in these geographical locations from where the samples were taken. So, for example, if you look at the far north clade or the far north region clade, there were 3 clades, I guess, and these 3 clades, these 2 clades from Atherton tableland, and these population were from Atherton tableland. They were sampled from there. And also the Innisfail clade, the samples, larvae and adults, were collected from Innisfail. That's how actually we put the geographical location of those clades. It is just a preliminary observation and analysis to show that there are multiple genetic groups. However, I understand that we need more in-depth analysis to actually confirm this one maybe in the later stage.
Okay, thanks, Emtia. Can I go on and ask Samuel a question? We can't.
Yeah, I was going to say, go and ask Samuel the question.
Yeah, thanks, Samuel. Look, you gave a great talk, and I just wanted you to give you an opportunity just to explain to me a little bit more about how these volatiles, I think you know this, but it just didn't quite come across, how the volatiles, once you identify them, how would they actually be deployed in a pest management package?
We will do that by deploying them in traps, attract and kill. So that we'll use them, yes, attract and kill.
Okay. Okay. So you just have a trap set up. The insects go, the adults go in there, and
Yeah,
then
it will attract them, because once we have them, we will attract them so that they don't even get a chance to go and lay. They will be attracted, then they will stay inside.
And so this is like a standard process in controlling other insect pathogens. And what sort of cost is this as compared to like an insecticide and its effectiveness, I guess?
Well, because this is environmentally friendly. This one is environmental friendly if you have to use an attract and kill. And although you may want to use, because you can design the trap site that you don't have to use mix any chemical insecticide. That is one option. Then there are other options where you would just add a little bit of the chemical insecticide as compared to if you are going to apply the chemical directly into the field.
And if I could just add a follow-up question to that, your data was quite interesting in that you found all these volatiles in the fig species, but you didn't find anything in sugarcane.
Yes.
So what's attracting the cane grubs to the sugarcanes then?
No, we did find some in sugarcane, but because it's not common. Like I said, the data is so huge that I couldn't present all the data. But the interesting one is there were some unique chemicals that were found in the most attractive species compared to sugarcane. Occasionally they will go to sugarcane, but they prefer the fig species because of the fact that they have a lot of volatile compounds emanating from them than the sugarcane.
Kevin, you want to add something, I suppose?
One of the things is that generally the beetles, the cane beetles, are not directly going to the cane for feeding and breeding. So, it's possible that for actually ovipositing or laying the eggs in the soil, there may actually be root volatiles there, which are released from the sugarcane, which then attract the beetles to go down and lay the eggs. So, we're talking about a different complexity of volatiles, really. For fig trees, it's just for feeding and breeding of the adults. But for the beetles to come in and lay their eggs, it could be quite a different system.
Thank you, Kevin. Samuel, there was a question from Hector Fleury. Could sugarcane be treated to produce volatiles that repel cane grub adults and prevent them from laying eggs in the crop?
Well, I think we will need further studies to confirm it, because even sugarcane has also produced other volatile compounds that were not found in the fig species. So perhaps maybe those compounds could be repelling the beetle, we don't know. But maybe if we have to do that, we have to further test that, develop individual treatment, or maybe get prototypes for each of these compounds to test them to see whether they will attract or they repel.
Thank you, Samuel. We've got another question, and it will be the last one because we need to wrap up. And it's for Emtia. So, seeing differences between I'm just reading the question, Emtia. Seeing differences between geographical population is normal and does not necessarily indicate any significant biological difference. Based on your phylogenetic tree, does the evidence suggest that there are two species? And what is the percentage COI sequence difference between the two currently well-identified species? And then the last question, so we have to be quick. How do you interpret the clade that is located far from the others from Mackay?
Okay. Very, very good questions, but I think I would need more time to actually answer all these. Well, for example, the last question was why that one clade was farther from the others, and this molecular analysis was done in the Irish lab, and I talked to the molecular specialist, and he confirmed that it is a distinct clade. It's not an outlier, but it's just a very distinct clade from others. So you can see there the branch is longer, and so that means historically somewhere it's connected, but the lineage is longer, and it means that this one is a genetically isolated population and found in Mackay region. That's all I can say from the phylogenetic tree. And for another question that has been asked about the sequence level, was it for COI, right? If I can remember.
Yes.
The COI. The COI sequence, I don't have the data with me. If you want to have more information on that, I can also discuss and give you the explanation on that. And of course, what is being said here, as I have to reiterate again, that we got multiple genetic groups based on the phylogenetic tree analysis, and it shows us there are distinct clades because all the clades are supported with a highly bootstrap values near to 70 to 100%. So obviously we can say they are separated from one another, but geographically, why we're saying that they're distinct from one region to another, because Bundaberg clades never intermingled with the Mackay clades. So obviously, based on that, we can say regionally there are separate genetic clades. But for more, we need more analysis to be done with the adults. I hope that answered the questions.
All right. Well, I just want to wrap up now. I really want to thank you, Emtia and Samuel, for presenting today. And I wish you a great continuation for this great work you're doing. I would like to thank everyone for their attendance today. And I hope you have enjoyed the last webinar for 2025. If you need more information on the webinar, please go and visit the event page of SRA website. Thank you again. I would really like to wish everyone a great weekend. I know it's not Friday, and because normally we do this webinar on the Friday, but and also I just want to wish everyone a really good Christmas break. Stay safe, and see you all next year for the 2026 SRA webinar. So Merry Christmas, everyone. Have a great weekend. Stay safe. Thank you.
Thank you.
Thank you.
Thank you.